Hello again. This is just the basics of energy for AP Bio. Our topics are going to be ATP, mitochondria and chloroplasts, cellular respiration, fermentation, and photosynthesis. First off, let's talk about the ATP molecule. ATP stands for adenosine triphosphate. The tri refers to the presence of 3 phosphate groups on this molecule. This is the molecule that's primarily used by cells as an energy storage molecule. And whenever a phosphate group is removed, the molecule becomes ADP and energy is released for cellular processes. To put that phosphate back on, you're going to have to use energy. So ATP functions much like a rechargeable battery in our cells. It allows us to store energy and release energy when needed. Mitochondria and chloroplasts are the main structures we think about with energy. I want to point out that they're only found in eukaryotes. If you guys remember that endosymbiotic theory states that these organelles used to be free-living bacteria, basically, and now they help their host with energy needs. Some support for this is that they have their own DNA, and in fact, that DNA helps encode for some unique proteins and enzymes that don't come from genes in the host nucleus. These organelles resemble bacteria in their shape, their function, and their reproduction. They actually divide by binary fission independently of the rest of the cell. Mitochondria are used in the cell to perform cellular respiration. They convert food molecules like glucose into ATP. Chloroplasts are used in cells to perform photosynthesis using chlorophyll. Basically, they collect light energy and convert it into food molecules. These processes take place in the cell membrane of prokaryotes. Even if you don't have mitochondria or chloroplasts, you can still perform these processes. And in fact, the fact that bacteria do it this way actually supports the endosymbiotic theory because mitochondria and chloroplasts perform these reactions inside what would be those bacterial membranes. All right, here's the cellular respiration reaction. We've got C6H12O6, glucose, plus 6O2, oxygen. And that's going to be converted during this process into 6 carbon dioxide molecules, 6 water molecules, and ATP. There's 3 main steps to cellular respiration. Glycolysis is going to break 6-carbon glucose down into 2 3-carbon molecules called pyruvate, and this takes place in the cytoplasm. The Krebs or citric acid cycle is going to break those 3-carbon molecules all the way down to carbon dioxide, and they're going to store that released energy for the next step. This takes place in the mitochondria. And then we're going to use oxidative phosphorylation, which is basically the electron transport chain and chemiosmosis, to use the energy from Krebs to make ATP molecules for the cell. This is also going to take place in the mitochondria. So let's look at that graphically. Basically, you can think of the slide as a cell, and the cytoplasm and the mitochondria are separate. The mitochondria are sitting in the cytoplasm. First, we're going to start with a molecule of glucose. That glucose is going to enter the glycolysis step. We're going to break that glucose in half, and we're going to feed the pieces into step 2, the Krebs or citric acid cycle. Then during that process, we're going to release some carbon dioxide, but mostly what we're interested in is taking that energy from those molecules over to the last step, which is step 3, the ETC and chemiosmosis. ETC stands for electron transport chain. This time we're going to take oxygen as an electron acceptor. We're going to put that into step 3. And when we do that, we're going to release a little bit of water, but more importantly, a lot of ATP. So this is how cells are going to convert glucose into usable ATP for cellular actions. Now, if it turns out that we don't have oxygen to complete the last 2 steps of cellular respiration, we're going to switch to a process known as fermentation. This is often called anaerobic cellular respiration because it's in the lack of oxygen. If there's no oxygen available, Krebs and ETC chemiosmosis are not going to occur, and fermentation is just going to be used to keep the cell going. The problem is that this process itself does not produce any ATP for the cell. The only ATP will be coming from glycolysis. If you have an organism that lives in an environment where oxygen is not available or has never really evolved to use oxygen, they usually have some other method to get around this rather than doing constant fermentation. But in organisms that are aerobic, like us, this fermentation step is kind of a backup plan. Now, there's 2 major types of fermentation. You'll hear about lactic acid fermentation, where the cell is going to produce lactic acid from those molecules made from the glucose. This is the one that we do. Usually when we hear about lactic acid, people talk about the pain and cramping of exercise. That's because you're not getting enough oxygen to your cells. So they're going into lactic acid fermentation. It's not literally burning your muscles, but it does lead to a cascade that does cause pain. Alcoholic fermentation is going to make ethanol and also release CO2. And this is the process that we've harnessed in yeast to make products like bread, beer, and wine. Now, photosynthesis, here's the reaction. It's going to look very familiar. This time we have 6 CO2, carbon dioxide, 6 H2O. And if you put those together in the presence of light, you're going to get glucose and 6 oxygen molecules. It's the same reaction backwards. But this time we're going to add energy instead of ending up with energy at the end. 3 main steps. The light-dependent reactions are going to collect light energy using the chlorophyll pigment, and we're going to convert that energy into ATP. The Calvin cycle is then going to take that ATP, and it's going to connect carbon dioxide molecules together to make short 3-carbon molecule chains. And then a final step that occurs in the cytoplasm is actually biosynthesis. And this is going to connect those 3-carbon molecules to make more complex molecules, the most popular example being 6-carbon glucose. So here, imagine the slide is a cell. We've got a chloroplast sitting in the cytoplasm, and the first thing that's going to happen is light is going to strike the chlorophyll pigments inside of that chloroplast. We're going to add a little water, and we're going to perform step 1, the light-dependent reactions. During this, we're going to make some high-energy ATP molecules, but we're also going to release a little oxygen as a byproduct. We had to break that water apart to get some electrons. So now that ATP is going to move over to step 2, the Calvin cycle. And during that step, we're going to take carbon dioxide, chain them together, and make some simple 3-carbon molecules that are kind of the precursor to sugars. Those molecules are going to go to step 3, biosynthesis. And the most famous molecule that's going to come out of this is going to be glucose. But those simple 3-carbon molecules could be made into many other things like amino acids. So photosynthesis and cellular respiration, I just want to kind of end with showing that this is really part of a bigger cycle. Photosynthesis is going to store energy. Cellular respiration is going to release energy. And the products of one are always the reactants of the other. So if you're a plant, if you're a producer, if you're an autotroph, you're going to do both of these reactions. You're actually going to store energy by taking light and converting it into food, and then use that food and take the byproducts to perform photosynthesis again and use that energy for yourself. If you're something like a heterotroph, if you're a consumer, you can't do the photosynthesis process. So you're going to rely on other organisms to do that for you. So you're either going to eat autotrophs or you're going to eat other heterotrophs that have eaten autotrophs. That kind of feeds into the whole ecological food web idea. All right, I hope that helped.